Last week, we introduced several different ATP power pump, P-class, V-class, and F-class proton pump. Okay, all of them only transport ion, pretty small. But now we move to the ABC superfamily. It's also belong to the ATP power pump, but it can transport not only ion, it can also transport a lot of different things. For example, amino acid, sugar, peptide, lipid, and other small molecules, including many types of drug. So ABC stands for the ATP binding cassette, ABC here. So this is pretty typical structure of the ABC transporter. It contains the two transmembrane domains, which will help them to embed in the lipid bilayer, and also contain the two ATP binding cassettes, which allow the ATP to bind there and do the hydrolysis, then release the energy for ABC superfamily to transport molecules. Okay, so now we want to explain why this transporter can transport different things, not only ion. Because first, this is a kind of typical ABC protein structure, or the blue color show the surface of the hydrophilic amino acid, and the yellow color show the hydrophobic amino acid on the surface of this ABC transporters. It have affinity with different either hydrophilic or hydrophobic molecules. And also it contains a very spatial structure. This cavity kind of extend halfway to the halfway of the lipid bilayer. Okay, so the molecule can bind, based on this, it can go into this cavity and bind with either hydrophobic or hydrophilic amino acid, help them to bind there. So just remember, the molecule binding in biology or either other system, it's not continuous bind there. It's not stay there. They will keep binding and they dissociate, bind and it dissociates. So you'll bind there and they dissociate. Then the ATP, it will use ATP will bind the ATP binding cassette, and the ATP hydrolysis will change the conformation, and it will change to the other conformation, then expose their cavity to the other side. Okay, so the molecule bind there, through the conformation change, they will kind of help them to expose to the other side. And this side is kind of open to the cytosol space or open to the extracellular space. Okay, that is why keep doing this through the energy from the ATP hydrolysis. They will bind with those molecules and expose to the other side. Then that's the way they keep pumping out specific molecules. And this is superfamily, which means it contains many members. So for example, ABCB1, ABCB4, ABCB11, I will not ask you to remember all of these members because it doesn't mean anything, right? Unless you study ABC superfamily, you need to remember, otherwise you don't, okay? So their original name is MDR, stands for multiple drug resistant transport protein. In the beginning, they had been found by a certain experiment. The researcher tried to incubate certain chemicals or toxic to kill the cancer cell. And they found normal cell can be easily killed, but the cancer cell cannot. And finally they found several proteins that will keep pumping out those drugs. That is why the drug cannot retain in the cell. That is why the cancer cell can resistant to the drug. So originally they has been named as their function multiple drug resistant transport protein because those superfamily, they are the guys keep using ATP hydrolysis as an energy to pump out the drug. That is why those cancer cells contain more ABC superfamily members and they can help them to resist the several drugs. Okay, but finally, because all of them kind of share the same structure, ABC cassette and the transmembrane domain, that is why they kind of be classified as an ABC superfamily. Okay, and just focus on here, ABCB4, which can export phosphorylthiodcholine into bile. Okay, this actually is this guy, which we introduced last week. Okay, during the phospholipid synthesis, it synthesized in the cytosol in the beginning. Then several key enzymes only exist on the ER membrane will take charge of the following step. And finally, those phosphorylthiodcholine, they will synthesize in the beginning, it was returned to the cytosolic leaflet. But this flip path, which actually is an ABCB4, will use energy from the ATP hydrolysis to flip this phosphatidylcholine from cytosolic leaflet to the exoplasmic leaflet. Okay, this ABCB4 is the guide to generate asymmetric distribution of the phospholipid. Okay, so how to confirm or measure the flip path activity of ABCB4? The textbook just give you this example. You can do this experiment to measure whether ABCB4 contain flip path activity or not. Okay, so first, they just collect the ABCB4 containing vesicle. How to do this? You just keep because ABCB4 will express on the cell plasma membrane, is also contained to the several secretory vesicle. Okay, so you can collect the median, okay, which contains secretory vesicle, and use antibody, ABCB4 antibody, which will bind to the ABCB4. Okay, and you can put your antibody to this is ABCB4 antibody, which can conjugate with certain kind of beads or agarose, and you can put on the column, the wall of the column. Then just collect those median containing the vesicle. Of course, they contain many different kind of vesicle. Okay, how to collect the vesicle contain ABCB4? You just pour the medians contain the vesicle to these columns. So ABCB4 antibody will bind with those vesicle contain ABCB4. Then you just wash out the other kind of non-related vesicle. Then finally collect those ABCB4 vesicle. Then you will get this, okay, in the Eppendorf. Then you just incubate those ABCB4 contained vesicle with certain fluorescence lipid. So based on those the principle that hydrophobic molecule like to interact with hydrophilic like to interact with a hydrophilic molecule, they gradually integrate into those vesicles. So your vesicle becomes kind of labeled with a fluorescence. Okay, so you can put to the plate reader or certain machine will give you a certain fluorescence number. Okay, then you get the vesicle contain the fluorescence lipid and also ABCB4. Then you just separate your reaction or your component to the two different tubes. The one tube you don't add ATP. The other tube you just add ATP to provide energy. So with ATP, theoretically, the ABCB4 will have energy to do the flipping. So you will flip the fluorescence, the lipid from the exoplasmic leaflet to the interior leaflet. Okay, but without the ATP, it cannot do anything. Okay, so now the key point is you use a chemical quencher, which is a kind of chemicals can bleaching the fluorescence. It will damage the fluorescence and make the fluorescence become dimmer or darker. Or you don't see any. If your fluorescence lipid exposed to those chemicals, they become dimmer. You don't detect any fluorescence anymore. Okay, but this quencher, which names we use dicyanide, which is a membrane impermeable chemicals. So it's only exposed to or damage the surface for fluorescence lipid. It cannot penetrate lipid. It will not damage the interior for fluorescence lipid. Okay, that is why after incubation with dicyanide, you will see only surface lipid become invisible. They become kind of has been quenched. Okay, but with the flip path reaction, because it will deliver more fluorescence lipid to the interior. That is why it can keep more fluorescence signal. Okay, so if you measure the whole reaction with their fluorescence signal in the beginning, all of them have the same fluorescence signal because you just separate the same reaction to the two different identical tubes. You add one with ATP shown in the red color, the other one do not add the ATP. So you will see the reaction with ATP keep the higher fluorescence signal. Without no ATP, without the ATP, you add quencher, it will reduce fluorescence more quicker or to the lower level. Okay, and finally, so this different theoretically is contributed by the ABCB4 fluid path activity. Okay? So, but why the final step, they still need to add detergent? Do you know why? We already got our answer, but why the next step, they still need to add detergent? Do you know why? Any volunteer? Yeah?
To extract the membrane.
Why they still need to extract the membrane protein? For the following other experiment or analysis, or you don't know? Okay, so because all of this just give you the number, you only got number from the plate reader. You need to prove those signals really come from the membrane structure. Okay, so you kind of do the positive control. You just add detergent, and it will destroy every kind of membrane-based structure, and all signal gone. That means this signal is not come from the non-binding fluorescent lipid or kind of debris or the garbage. It's really come from at least the membrane structure. Okay, just double confirm. Though you are not, this is not imagination. It's really come from what you design. Okay, that would be their experimental design. So that is why it becomes this, and it would damage any signal or eliminate any signal from the membrane-based structure. Okay. So next, ABC superfamily layer will be the other famous members called cystic fibrosis transmembrane regulator, or CFTR. Just remember, not all ABC superfamily are pumper, okay, are not pump. Okay, this guy is ion channel, although it needs energy. Okay, it needs energy to open the ion channel. It doesn't need energy to deliver molecule. Ion channel doesn't really drive the delivery. It only opens the gate. Okay, so this also is contained 2 ATP binding cassette and 2 transmembrane domain. In steady state, without the ATP, it's kept in the closed kind of conformation. But once you bind 1 and 2 ATP, they will form such a heterodimers, and then with this conformation, they will open the ion channel. The chloride will still follow its electrochemical gradient to transport in or out. Okay, so after the ATP deassociate or hydrolysis, it will go back to the closed conformation, then stop anything kind of chloride delivery. So why it's called the cystic fibrosis transmembrane regulator? Because if mutation happen to that protein, will cause the one disease called cystic fibrosis, okay, which contain really, really strong kind of severe inflammation in the body. So why? This is the kind of epithelial cell in our airway. You will see in our airways, the surface contain many multicilia. Just check this video. This is the cartoon, this is the real video. You will see many cilia that keep moving and generate the flow, okay? In our airways, those flow can help us to kind of generate the flow and help us to remove the mucus. The mucus contain many dead bacteria or pathogens. It will help us to remove those mucus, okay? So, but you need to hydrolyte those mucus, otherwise those mucus will become really sticky. It cannot be easily moved, okay? So how to hydrolyte those mucus? You need to use a CFTR. That's the ion channel we just introduced in the previous slide. It will release the calcium chloride to the airways lumens, okay? And the chloride, because the water is not charged molecule, but water is a polar. It still have a kind of more negative end and a more positive end, right? H2O, like right? It will be more negative end and the positive end. So chloride will also kind of bind with the water molecule. So it will kind of attract more water in these lumens, okay? So that is why contain more water, it will be easier kind of to deliver such a flow and remove the kind of bacteria and the pathogen. But if mutation happen to the patient, cystic fibrosis patients, the the CFTR protein cannot normally go onto the surface, and the chloride cannot really deliver to the airway lumens. So you cannot keep the water here. So that will make a mucus super sticky, super sticky. And many bacteria retain there and even form the biofilm. So even those multicellular, they keep working really hard, they cannot move that mucus. So it will trigger really severe inflammation due to the mutation of the single protein. Okay? Of course, once we know which mutant caused this disease, people now have many different strategies to try to do kind of therapy. For example, we have a CRISPR-Cas, we have several vehicles can deliver those components. So for example, last week we introduced a liposome, but recently those LNP will be more popular. It's kind of lipid-based nanoparticle. It will mix with those DNA. That's the way they deliver the vaccine, mRNA vaccine. Moderna will use an LNP, okay? And the guy got the Nobel Prize? Did he? No, not that guy, okay, no, okay. So they can use this to deliver into the they cannot kind of directly try this to the human body, so they just try to the human cell, and F508 deletion mutation happen to the 508 amino acid. That will kind of trigger the deletion, early deletion of the CFTR. Happens to the 70% of all CF patients. So you can change this stop codon and make it kind of express the patient cell express normal CFTR or full-length CFTR. It can really help to kind of improve the symptoms. Okay, of course, to the mouse, it contains different mutant. It's also the case can help mouse to kind of cure such a diseases by doing this. Okay. So let's just give you the example of how ABC superfamily can use energy from the ATP hydrolysis to transport different molecules. Okay, any questions so far? No? Okay, so move on here. You will see this is a water tank. It contains two kind of different areas. So it was separated by the membrane shown here. In the figure A, there's no difference in the concentration of the cation and the anion between two sides, okay? But if this membrane allowing permeability of only sodium will be more sodium transferred from this side to the other side, then after a while, will be more a cation accumulate to the left side, okay? And there will be kind of more negative charge in the right side, okay? So such a voltage difference will generate, we call the potential, because the positive molecule would like to transfer from this end to the other end. Negative charge molecule would like to transfer from this end to the other end. It's kind of have a potential energy stored there. We call this potential, happen to the membrane called membrane potential, okay? So it's just like a battery. It's contain the energy, contain potential there. Once you kind of use that battery, it can use the electric, right? Okay. This also happens when the membrane allowing the potassium to transfer from this side to the other side will also generate the voltage difference happen to the membrane will generate membrane potential. Okay. Cell can use that to do several jobs through this energy. So it's also the case to the cell membranes. This is a plasma membrane, there's an extracellular space and the cytosol. There will be more cation on the surface and an ion in the cytosol. Okay, that is why the human being always be more negative, right? Just joking. Okay, so why cell can achieve this or accumulate more negative charge or an ion inside the cell? Because there are many different opening channel, like a potassium channel or different channel, but there are more potassium channel open on the cell surface. So they allow a lot of potassium positive charge ion go into the surface. Okay, that is why they will contribute to this membrane potential. Okay, we call this because it's the open of those potassium channel is not depends on any activation or regulation. It always happens. So even in the resting state. So that is why we call this a resting potassium channel. Okay? So next question is why we call that potassium channel? Because it's only allowed potassium go across that channel, right? We don't call that, it should not allow the sodium or calcium or chloride to penetrate. That is why it's called the potassium channel. How can it achieve such a selectivity? Okay, of course, it's not depends on the charge because the only transport allow the potassium, not the sodium or chloride. How can it achieve this? That will be the next issue we'll try to introduce. So we use this resting potassium channel as examples. So this is a monomer of the resting potassium channel. It's contains 2 transmembrane domains, and it also contain this beta strand is we call the P segment. And this will be the the function will be the selectivity, the filter. Is the region achieve such a potassium specificity, okay? So this is monomer, and the resting potassium channel will assemble as a heterodimer. This is the top view, okay? There is 1, 2, 3, 4. And they will face their selective filter to the center or the way the ion will go there, okay? So the ion will touch those selective filter and go to the other side, okay? How can they separate potassium to the other cation? First, as I mentioned before, the ion in the aqueous solution, they will try to bind with water. Water doesn't have a charge, but they still have a more two end. It's a polar molecule. It still have a negative end and a positive end, okay? So those cation will bind to the negative end of the water. So it will bind like this. And that will be they can form the water shell on those ions. So the surface of the potassium is a little bit larger than sodium. And the surface of potassium is good for 8 water molecule binding. But the sodium surface is only good for the 6 water molecule binding. 8 versus 6, okay? It doesn't matter in the water solution because it's just free moving environment. But once they approach to the resting potassium channel, this selective P segment, the selectivity, the filters, their carboxyl group will mimic the water molecules and provide a binding site for the potassium or sodium to bind. Okay, they don't need to bind the water molecule. When they approach to the selective filter, they will bind with those carboxyl residue from their P segment. So when they bind to this is in the aqueous solutions, it will bind with 8 and the 6 water molecule. But once they approach to those pore, the size, the distance between those carboxyl group, it's totally identical, fit the potassium ion. Okay, so they can feel nothing different. They just approach there and still bind with this. They don't exposed to any energetic invariable conditions, and they will keep binding, and go to the next step until they leave those selective filters and bind to the other water molecule. Okay, but for the sodium, it's smaller. It will bind with these 6 water molecules, and they feel pretty comfortable about this. But once they approach to the resting potassium channel, it will only bind to the one side. It's smaller, so it will expose their charge here. It's not energy favorable conditions. So that is why they will go back to bind with the water molecule. That is why it's prevent the sodium to go to the deeper of the resting potassium channel. Okay, that's the way resting potassium channel can only transport with the potassium, but not the sodium. This is only the case for the resting potassium channel. Other ion channel also use different mechanisms to achieve, such as selectivity. They can use a side chain, they can use the water shield mechanism, they can use a kind of such an interface to mimic their favorable condition in the aqueous solution, so to allow the specific molecule to go across, okay? So patch clamp is a method you can use to measure the ion movement through the single channel. So you can use a capillary to form to act as an electrode, and put one electrode on the cell membranes. And of course, there are many different ion channels on the cell membrane. You just put the capillary electrode to the cell surface and using a little bit, suck a little bit. So you will generate a closed environment here. And also put the other electrode into the cell. It's used as a reference. So you can measure the voltage difference. And also any ion go through this tip of the electrode. It's about the 0.5 micrometer. It's pretty tiny. It's only covered the area good for the one or few ion channel, okay? So based on this, you can add drug or to calculate any charge voltage difference in these environments, okay? Then you know the ion channel activity, whether it's any ion go across that channel or not, okay? So this is the video.
Small opening is used to make tight contact with a tiny area or patch of neuronal membrane. After the application of a small amount of suction to the back of the pipette, the seal between pipette and membrane becomes so tight that no ions can flow between the pipette and the membrane. Thus, all the ions that flow when a single ion channel opens must flow into the pipette. The resulting electrical current, though small, can be measured with an ultra-sensitive electronic amplifier connected to the pipette. This arrangement is the cell-attached patch clamp recording method. A record of the current flowing through a single ion channel reveals when the channel is in an open or closed state.
When you open, you would show the lower voltage level.
Are called microscopic currents, to distinguish them from the macroscopic currents flowing through a large number of channels, distributed over a much more extensive region of surface membrane.
So you can measure in the steady state, whether in the certain time, how frequent the ion go across that ion channel, okay? It will be and they're pretty similar. And you can add a different drug inhibitor to see whether you can block those ion channel by doing this. And also, how can you really confirm this is come from? Because you cannot really see the ion channel, right? How can you really confirm those data come from the single ion channel or multiple ion channel? It depends on these tips, okay? If you measure the single ion channel, it will only go certain level. But if the data come from the multiple ion channel, it will go deeper, okay, in the steady state. So you just keep measure the background ion channel activity, you will know whether now the area you touch covered by 1 or 2 or even 3 will go deeper, okay? So once you so you need to move to the other space until you can get the single level, single ion channel data, okay? Okay, but the other question is, you cannot see the ion channel. How can you really make sure the activity you measure is come from the one you are really interested in? For example, if I want to measure the CFTR ion channel activity, how can I make sure? I cannot see that. Okay, how can I make sure the one or the data I got really come from CFTR? So there will be several ways. First is frog oversight expression system. Basically, you need to find a system which do not express or just express really low endogenous ion channel. Okay, the one will be the frog oocytes. Before fertilization is in the static resting state, they do not express any ion channel. Okay, so you can use a microinjection to inject the mRNA of the ion channel you want to study. So after 24 to 48 hours, the oocyte will express that ion channel on the surface. So now you do the patch clamp to that frog oocyte. Any signal you got is all come from the ion channel expressed from the mRNA you inject. Okay, that will be kind of the one system. And usually people recently they most usually use the Ch10&11 cells as a model to measure the patch clamp because the two reasons. The first, there are multiple reasons. First, Ch10&11 cell is super easy to culture, okay, and the second, it's really easy to deliver DNA into the Ch10&11 cell. You always get really high transfection efficiency. You can put the DNA and then express to almost every cells, okay? And the second, it has low endogenous ion channel activity. So mainly the signal you come from is not 100%, okay? But the mainly data you come from the Ch10&11 cell, if you can measure anything, it's come from the signal activity from your exogenous ion channel. Exogenous is mean the DNA you artificially deliver from the outside. Okay, endogenous is the original, the cell contain the original the gene expression. Okay. Yeah. So Ch10&11 cell will be the one to give you that serve as that model organism. Okay. So in this section, we just introduce. So here we just introduce the resting potassium channel will contribute to the membrane potential, and how the resting potassium channel is possess a P segment and can achieve the ion selectivity. Okay, and the patch clamp can permit a measurement of the ion movement through the single channels, and to get the oocyte expression assay, and the patch clamp can use to characterize the ion channel you want to study. And of course, the Ch10&11 cell will be have been commonly used for the patch clamp in mammalian systems. Okay. Okay. So next we move to the co-transporter. As we introduced last week, there's two types of the co-transporter. Depends on the direction they transport. It will be the sympater and the antipoter. So for the sympater, it will transport the pink or the red molecule through their electrochemical gradient, follow its electrochemical gradient, and use this as energy to transport black one against their electrochemical gradient. And they transport in the same direction. But the antipoter will use the energy to transport the red molecule, follow its electrochemical gradient, and use this energy to transport the black one against its electrochemical gradient through the different opposite direction. Okay. So for the sympater, this will be the one example. So 2 sodium, 1 glucose sympaters. So it contain the 2 sodium binding site and the 1 glucose binding site. And then the sodium will bind to those binding site, and the glucose will also bind here. And then it will use this sodium follow its electrochemical gradient to transport to the other side. Binding of this will change their conformation. Then it change to the second conformation. Then the affinity of the calcium binding site will become weaker. That is why we release the calcium to the other side. Okay, and in this conformation, it will also the glucose binding affinity will also become weaker. So that is why we release here. And releasing of those 2 molecules will kind of turning back is a conformation to the conformation in the beginning. Okay, then they have a strong binding affinity with the sodium and the glucose. Keep doing this, they will keep use the sodium as an energy to transport glucose to the other side. Okay, and next question is why they need to co-transport 2 molecules? They have sodium binding site, they have a glucose binding site. Is it possible they only transport sodium, or solely transport sodium, or only transport glucose? No, they need to co-transport 2 molecules simultaneously. Okay, why? Just give you the example, which is a sodium 1 leucine sympaters. Okay, so two sodium, one leucine sympoters. So this sympoter will transport leucine and the sodium together at the same time. Because the sodium, as I mentioned before, it will bind with the 6 water molecule, right? So once they approach to this sympoter, 5 binding site will kind of contribute from the carboxyl group of those sympoters, and then 1 binding site will come from leucine. Okay, so only when the leucine approach and this sodium approach together, it will become the energy favorable condition. Then they will become really comfortable here. Then the sympoter will be able to transport them together. If they don't come together, nothing will retain there, and they cannot transport together. Okay. So then we'll give you the example of the antibodies, which is the AE1. It will transport, as you can see here, will transport the molecule to the opposite directions. So this AE1 will help the red blood cells to do the different job in the different system. For example, in the lung, it will help the red blood cell to absorb more oxygen and release CO2. But in the body somatic regions, it will release oxygen and uptake the CO2. Okay, the same system can do the opposite reaction. It's driven by those antipoter AE1. Okay, so let us start from here. The CO2 uptake. CO2 in the gaseous form, CO2 cannot really dissolve in the water. So basically, you just go in and go out. You cannot really retain CO2 in the red blood cell or in your body. That is why drinking Coke will make you burp, right? You cannot retain the CO2 in your body. You just release them through burping. Okay. So the CO2 how to keep the CO2 in the water? The carbonic anhydrase, this enzyme will combine CO2 with OH group and form the HCO3. HCO3 will be the form can really dissolve in the water solution. That is the way they can keep the CO2 component as a HCO3. Okay, but the AE1 will transport a lot of HCO3 out to the cell and use a chloride to transport chloride as an energy to transport a lot of HCO3 to trigger such an influx, exflux. Okay, just go out to the cells. So the CO2 will keep doing uptake here by keep pumping out to the extracellular space by the AE1. And this OH comes from the water splitting. So the water will split OH and the proton here. And the proton will bind to the other important protein, hemoglobin. The one residue is histidine residue of the hemoglobin. It will bind here, will change their conformation. And such a conformation will lower their affinity with the oxygen. So that is why they will release oxygen. But when they approach to the lung, again, carbonic anhydrase will split HCO3 to the CO2 and the OH. So CO2 will be easy to leak out from the red blood cells because it doesn't dissolve in the water. And this OH and the proton will form the waters. And the associated proton from the hemoglobin will change their conformation and then will become the higher affinity with the oxygen. That is why they can uptake the oxygen there. And they will also transport chloride and HCO3 to make sure the whole reaction will go through the opposite kind of direction. Okay, that is why the same system can do the totally opposite reaction. Okay. And also in the vacuum, many different transport. You will see a lot of antipoda here. So in the vacuum, in the plain vacuum, they will store many different nutrients and the essential molecule in the vacuum. Okay, so it's driven, of course, it's driven by the energy. So the several proton pump protein, they will use ATP energy from the ATP hydrolysis. It pump a lot of proton here and generate electrochemical gradients. And then chloride will go in, and that is why they bind with the HCl and become more acidic. And also this electrochemical gradient will also attract other NO3 or other essential molecules. And many antipoda will use this electrochemical gradient to release the proton and use this as an energy to transport sodium, chloride, and the sucrose essential molecule in the vacuum. That's the way plan to transport specific or essential molecule in the vacuum. Okay. So that would be kind of we use several examples to show you what's the symphoter. They co-transport different molecule in the same direction, and the antipoter they co-transport different molecule in the opposite directions. So for the sympoters, we just use a 2 sodium 1 glucose symphoter to co-transport sodium and glucose in the same direction. We also use leucine and the sodium, 2 sodium and 1 leucine sympoters as an example to show you why they need to transport different molecules simultaneously. And the AU1, as an example, we showed you in the red blood cell, they co-transport HCO3 and the chloride in opposite direction. So by doing this, they can, in the same system, they can do the really opposite job, okay? And the different transport protein will enable the plant vacuum to transport, accumulate, metabolize, or essential ion in their vacuum, okay? That would be the introduction about the symphoter and antipoters. Okay, so in the final section of this chapter, we just give you the 3 examples, the real example, how the different transporter, ion channel, ATP power pump co-work together to make sure the physiological process in our body can really properly conduct it. Okay, so this is the intestine. Okay, when we eat food, it will be digested by our digesting system. And finally, digested food will go to the lumen of the intestine. Okay, so this is the intestine lumen. So let us enlarge here. So this will be intestine lumen. So the digested food will be here. So the mission of those intestine will try to uptake nutrient from the food across this layer of the cells. Okay, it's the epithelial cell, intestine epithelial cell, and uptake the nutrient and across this and go to deliver those nutrients, go to the circulation system. Then the nutrient will go to the other part of our body. That is why our whole body can receive the energy or nutrient from the food. Okay, so let us enlarge here. Those cell, intestine epithelial cell, this is the lumen, they try to uptake more nutrient from this side to the circulation system. Okay, so to do this is also need energy. They use this sodium, potassium, ATPase, use the energy from the ATP hydrolysis to transport sodium and the potassium and generate the sodium electrochemical gradient of the sodium here inside the cell. So this sodium and the glucose symphoter will use this energy, just transport sodium, follow its electrochemical gradient, and you use this energy to uptake glucose into the cell. Okay. Then the GLUT2 will transport glucose to the other side, to the circulation systems. Okay, and those potassium channel just try to balance this kind of electrochemical gradient. Okay, that's the way why the intestine epithelial cell can keep uptake the glucose and the transport to the circulation system. In the stomach, okay, this is the stomach. The lumen of the stomach is really acidic. Okay, it will help to kind of many enzymes to digest the food. They like to trigger the enzyme reaction in the acidic environment. So as you can see here, stomach lumen is pretty acidic. The pH is 1, super acidic. Okay, it's contribute from the stomach wall, epithelial cell on the stomach wall. So they keep pumping proton and the chloride. That's why they can make this environment super acidic. Okay, so the potassium and the proton and the potassium ATPase will use the energy from the ATP hydrolysis to transport proton here and the potassium here, and it will balance. Potassium will balance by potassium channels, and those proton come from the water splitting, and the OH will combine with the HCO3 by carbonic anhydrase. And the AE3 antipoters will transport HCO3 and transport a lot of chloride in the opposite directions. And those chloride will transport to the lumen through the chloride channel. Okay, by doing this, a proton will bind with the chloride and become the HCl. That is why it becomes really acidic. Okay. And the third example is just 2 kinds of cells. One is osteocyst. Okay, osteocyst here, and the other is osteoclast. there are two kinds of cells. Osteocysts will help you to generate more or grow bone, more bone. Osteoclasts will help to, they will eat bone, okay? Coordination between these two types of cells will help you to shape the beautiful or proper bone, okay? So here they just kind of focus on how these osteoclasts can digest the bone. First, they need to attach to the bone and make a closed environment. They use a really strong connection here, okay, and make this a closed environment. And they use ATP hydrolysis proton pump and use energy from the ATP hydrolysis to pump a lot of proton here, and also use this chloride channel to transport chloride here. And HCl will make this environment super acidic. That's good for the enzyme reaction to digest bone, okay? And the chloride will come from the antipodes. Of course, those will be the kind of pretty similar mechanism. CO2, the water splitting here, and the proton will come from here. And the OH will combine with the CO2 to become the HCO3. And the antipode will transport HCO3 and using this as energy to transport chloride here. That is why finally they will make this environment super acidic, okay? That would be the kind of few example how these different transporter or ion channel or ATP power pump co-work together to conduct different physiological reaction.